Hello, everyone. Welcome to Brain Blitz Audios. In this episode of NEET preparation, we'll be dealing with previous year questions of biology in medical entrance examinations in India. The chapter that we're dealing with is biomolecules. This is the 9th chapter in the grade 11 syllabus for CBSE. Let's begin with our first question. So the question is asking us to find which of these is a derivative of an amino acid: epinephrine, estrogen, progesterone, or all of these. Estrogen and progesterone are steroid hormones. Now, what that means is that they're derived from sterols, which are fatty acids. So these are basically lipids instead of amino acids. So options B and C are incorrect. Now, since B and C are incorrect, we can safely say that option D is also incorrect. Because if option D was correct, then all of these would be derivatives of amino acid. But estrogen and progesterone are steroids. So option D is also incorrect. The right answer is option A, epinephrine. Epinephrine is derived from the amino acid tyrosine. So epinephrine is a derivative of the amino acid tyrosine. Let's look at our next question. The high energy bond of ATP are present in between carbon-carbon, carbon-oxygen, carbon-nitrogen, oxygen-phosphorus. So in order to generate energy, ATP is changed into ADP plus phosphorus. So one bond with phosphorus is getting broken. So the only bond with phosphorus in it is option D. So this bond has the high energy, and it changes ATP into ADP plus phosphorus. Why are the others wrong? If we break a CN bond, it is basically leading to change in proteins. And option B, CO, is the splitting of carbon oxides to form oxygen. And the CC bond is broken in order to break polymers. So when you break polymers such as plastics and Teflon, etc., so you break the CC bonds. So option A is incorrect. The right answer is option D, oxygen phosphate bond, oxygen phosphorus bond. So this is known as the phosphate bond. And when you make a phosphate bond, you get high energy. That's why ATP is known as the energy currency of the cell. Next question. Who coined the term zymase? Is it A, Pasteur, B, Buchner, C, Kuhne, and D, Sumner? Let's look at each of our options. Sumner proved that enzymes are proteins. So therefore, option D is incorrect. Option C, Kuhne. Kuhne is credited with discovering the enzyme trypsin. So option C is incorrect. Option A, Pasteur, is associated with microbiology. Processes such as pasteurization and disinfection come from Pasteur. So option A is incorrect. The right answer is option B, Buchner. He used the term zymase to refer to a complex of biocatalysts. And he found these in yeast while undergoing alcoholic fermentation. So yeast, when undergoing alcoholic fermentation, gives out a complex of biocatalysts, which Buchner renamed as zymase. Next question. Apoenzyme is A, protein, B, lipid, C, sugar, D, vitamin. Now, we know that enzymes are proteins, and the enzymes are further divided into apoenzymes and cofactors. And we also know that these cofactors are usually metal ions. So on that note, let's look at the following options. D vitamins are present separately in the body. So option D is incorrect. Option C, sugar. The apoenzyme does not contain sugar. Sugar is found in glucose, starch, carbohydrates, etc. So option C is incorrect. Option B, lipids. Lipids are components of body fat. So lipids make up fatty acids and oils, etc. So option B is incorrect. The right answer is option A, protein. Since enzymes are proteins and they're composed of apoenzymes and cofactors, cofactors are metal ions, so apoenzymes have to be proteins. So option A, proteins, is correct. Next question. Proteins are polysaccharides, polyamides, polynucleotides, polyglycol. And if you remember, proteins are derivatives of amino acids. So now let's look at each of our options. Polysaccharides is option 1. Examples of polysaccharides are sugar, starch, cellulose, carbs, etc. So option A is incorrect. Option D, polyglycol. Polyglycol is present in substances such as glycerol and fatty acids, oils, etc. So option D is incorrect. Option C, polynucleotides. Polynucleotides essentially are DNA and RNA. These constitute the genetic material. So option C is incorrect. The right answer is option B, polyamides, because amino acids are converted to amides, and these amides are present in polymers, so polyamides, to form proteins. So option B is the right answer. Next question. Which of the following gives the Fehling's test? Pectin, sucrose, cellulose, glucose. Now, in order to give the Fehling's test, the substance has to be reducing. It has to have a functional group that is reducing. So pectin is sugar acid of galactose, which is non-reducing. So option A is incorrect. What about option B, sucrose? Sucrose is an example for a non-reducing sugar, which is why it is sort of used as table sugar. So option B is incorrect. What about option C, cellulose? Cellulose is again a polysaccharide which is non-reducing. So option C is incorrect. The right answer is option D, glucose. And the reason why glucose gives the Fehling's test, because the free aldehyde, that is CHO group, gives the Fehling's test. It undergoes reduction to give Fehling's test. So that is the right answer, option D. Glucose gives Fehling's test. Next question. Nicotinamide is synthesized in our body from A, tryptophan, B, tyrosine, C, valine, D, alanine. So nicotinamide is an enzyme which is present in short quantities in the body. So we are to find out which of these is used in synthesizing nicotinamide. Is it A, tryptophan, B, tyrosine, C, valine, or D, alanine? Let's look at each of our options. Alanine gives you strong immunity. So option D is incorrect. Option C, valine. Valine is found in muscles, so it is used for maintaining muscles. Option B, tyrosine. Tyrosine synthesizes epinephrine. So option B is incorrect. The right answer is option A, tryptophan. Tryptophan creates nicotinamide. It is used in synthesizing nicotinamide in our body. Next question. An example of competitive inhibition of an enzyme is the inhibition of succinate dehydrogenase by malonic acid, cytochrome oxidase by cyanide, hexokinase by glucose 6-phosphate, carbonic anhydrase by carbon dioxide. So let's look at each of our options. Options D and C. Carbonic anhydrase by carbon dioxide, hexokinase by glucose 6-phosphate are non-competitive because this is basically a feedback mechanism. So in both cases, so if there is a lot of carbon dioxide in the body, the carbon dioxide itself would start inhibiting carbonic anhydrase. So there is no scope for competitive inhibition. It's the same for hexokinase. When glucose 6-phosphate, which is similar to fructose 6-phosphate, inhibits hexokinase, so that is also another feedback mechanism because hexokinase is used for creating glucose 6-phosphate. Options D, C are incorrect. Option B, cytochrome oxidase by cyanide is non-competitive inhibition because it has a separate allosteric site where cyanide binds to cytochrome oxidase. So it changes the active site, so there leads to problems. So that's why people die of cyanide, because cyanide inhibits cytochrome oxidase and it's non-competitive, so the person dies immediately. So option B is incorrect. The right answer is option A, succinic dehydrogenase by malonic acid. So the malonic acid, it inhibits succinate. So it inhibits succinic dehydrogenase, so it inhibits succinate production. So when there is higher amount, then the effect of malonic acid is negated. So that is option A is the right answer. Next question. Which of the following set of 3 items are not true as each set belongs to the category mentioned against them? So if, for example, A is lysine, glycine, and thiamine, then what the question says is that the set are all amino acids. We need to find out which of them is not true. So we need to find out which of this is true and hence prove that the others are not true. So here in option A, we have lysine, glycine, and thiamine. And it's given that all 3 of these are amino acids. However, thiamine is actually a nitrogenous base. So therefore, option A is incorrect. So here the question is asking us to find out which of these is true because there are 3 of them incorrect and 1 is correct. So next option, B, myosin, oxytocin, and gastrin. Now these are hormones, which is what is given. But myosin is actually muscle protein and not a hormone. So option B is incorrect. What about option D, optic nerve, oculomotor nerve, and vagus nerve? All of these are given as sensory nerves, but the oculomotor is a motor nerve and the vagus nerve is a mixed nerve. That means it can undergo both sensory and motor functions. So option D is incorrect. The right answer is option C, renin, helicase, and hyaluronidase. So all of these are enzymes, and when you look them out, renin is an enzyme to catalyze milk, helicase is another enzyme in DNA, and hyaluronidase is again another enzyme. So all three of these are enzymes, so this is true. Option C is the right answer. So the question is asking us to find out which of the following sets of three items are true, as each set belongs to the category mentioned against them. And after detailed analysis, we find that option C is the only option which matches this. So this is the right answer. Next question. The figure given below shows three velocity substrate concentration curves for an enzyme reaction. What do the curves A, B, C depict respectively? So we have four options. So let's look at the graph in general. So the graph has initial velocity V0 and substrate concentration S. So we have the quantity Vmax in the case of initial velocity. And then we have another quantity that's Km, Michaelis constant, in the substrate concentration. So therefore, this is of a graph of enzymes, the functioning of enzymes. So the first graph, that is A, will always be normal. So this is normal enzyme function. Now, if you look at the second one, B, you see that it's extended towards the right. And so it takes a greater amount of substrate in order to reach Vmax. So that scenario is found in competitive inhibition. So that means it has a greater Km, and that also means longer time to achieve Vmax. However, if you look at graph C, you see that that the graph never reaches Vmax. So that means the enzyme is not working. And this situation happens in non-competitive inhibition. So A is normal enzyme, B is competitive enzyme, and C is non-competitive enzyme. So therefore, option A is the right option. So that is the right answer. So now the next question. Which of the following contains beta-1,4 linkage? Is it A, maltose, B, sucrose, C, lactose, D, fructose? Let's look at each of our options. Maltose contains an alpha-1,4 glycosidic linkage, which is not beta-1,4 linkage. So option A is incorrect. What about option B, sucrose? Sucrose again has a glycosidic linkage between the first carbon of glucose and the second carbon of fructose. So option B is incorrect. What about option C, lactose? Lactose undergoes condensation reaction to form a combination of galactose and glucose, which has beta-1,4 linkage. So option C is the right option. And option D, fructose, is incorrect because fructose is a monosaccharide. So there will be no linkages between two monosaccharides in a monosaccharide because a monosaccharide is just one. So option C, lactose, is the right answer because it undergoes condensation reaction to form beta-1,4 linkage between galactose and glucose. Our final question. Which of these statements is true? Adenine has 4 nitrogen atoms, cytosine has 3 nitrogen atoms, guanosine has 3 nitrogen atoms, uracil has 5 nitrogen atoms. Let's look at each of our questions. So if if you look at the structure of uracil, you count 2 nitrogen atoms. However, the question says the uracil has 5, so option D is incorrect. What about option C? Guanosine has 3 nitrogen atoms. The structure of guanosine, when you count it, you find 5 nitrogen atoms, so option C is incorrect. What about option B? Cytosine. Cytosine, it says, has 3 nitrogen atoms, which is incorrect. It only has amides. And therefore, that proves that option A is the right answer. Adenine has 4 nitrogen atoms because adenine is a purine, and purines have a 9-membered ringed compound, and the rings itself are twice. So there are 2 rings and 9 members in a purine compound. And so adenine will always have 4 nitrogen atoms. So that was the final question, and it concludes this episode of NEET preparation. There are a lot of other questions as well regarding the chapter biomolecules, but due to shortage of time, we'll be stopping here. However, you can find the links for our other videos on NEET preparation in the description down below. And of course, don't forget to subscribe to our channel, Brain Blitz Audios, to have access to a lot of free educational content such as learn awards, mind maps, NEET preparation, CBSE board exams, etc. So until the next episode, take care, stay safe, ta-ta for now.